So in their publication, Watson and Crick noted, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So do we remember who Watson and Crick were? They put together the ball and stick model of the secondary structure of DNA. They are credited with figuring out the structure of DNA. So they proposed this semi-conservative replication, saying that you have one parent molecule and it separates and uses the original or parent strand as a template for the two daughters. So what you're left with is a DNA molecule that has an old strand and a new strand. But that wasn't the only way it could have been. So there's also conservative and dispersive replication that was put forth. So we had to come with a way to figure out which way is it actually doing. So semi-conservative is what they put forth. You have the parent molecule here. It's all pink. The daughter molecule is going to contain a parent strand and a daughter strand, or an old strand and a new strand. So you see an old pink strand and a new blue strand. The conservative model says that you start with this parent strand that's in pink again, and you use it to create a new daughter molecule. So the entire daughter molecule is two new strands, and the old parent molecule is still two old strands. So one new molecule, one old molecule. Two new molecules that have half old and half new. Then the dispersive says you go into the parent, you cut it up into pieces or segments, and then you fill in the blank with a new one. Okay, so that essentially you get two molecules that has some old parts and some new parts in the same molecule. So that's the idea for this dispersive model. So we had to figure out which one is correct. So is what Watson and Crick put forth actually how DNA replicates, or does it use one of the other two models that are possible? So to figure this out, Messelson and Stahl did an experiment. So they grew bacteria in N15, okay, so that the DNA that was made has this heavy nitrogen in it. And so when you centrifuge it, you see, hey, I get a heavy band. I know that all of this DNA is heavy. Now I'm going to transfer those cells that have the heavy nitrogen into light nitrogen and let them grow. So now what I get is this hybrid molecule that has essentially, it looks like some heavy and some light. Then I'm going to grow it again and see what happens. And when I grow it again, I get a light band and a heavy band. And from this, they were able to figure out that it is indeed semi-conservative. So now you're sitting there probably like, huh? What? How? So let's look at this again. So if it was the conservative model, then you would get a completely heavy strand and then a completely light strand. So here in the end, they would have had a light and a heavy. They didn't see that. So we could say, okay, it's not conservative. Now they got half heavy, which means it could have been semi-conservative, it could have been dispersive. So after they did it for several generations, you see you're left with a light band and a half heavy. So that's saying it has to be, this is what's going on. So we get light bands, we get half light and heavy. Here, we would expect to see varying degrees of it still containing heavy molecules because we know that we're still cutting it up and filling in the blanks. So each molecule that we make are still going to have portions of that heavy in it. So because we didn't see that, so we would have seen, let's go back to this one. We would have seen somewhere in the middle where we have varying degrees of weight because we have varying degrees of heavy still in our molecule. So this was how they ruled it out. I hope that makes sense. If not, we can talk about it a little more in class. But essentially, they grew DNA with heavy nitrogen so that they're making heavy strands or heavy molecules of DNA. Then they switched it to light nitrogen so that they're making lighter. And so if it was conservative, which is one completely old molecule, one completely new, they would have seen one heavy molecule because that's the old one, and one light because that's the new. But they've seen a half heavy. Half heavy means it's light and heavy in the same molecule. So because they've seen that, it could have been semi-conservative or dispersive. So then they had to grow it out some more And when they did that, they seen some bands that were completely light, which says, hey, this is semi-conservative. We're getting half heavy, which has old and new, and we're getting completely light, which is new. If it was dispersive, they would still see that heavy lingering because the molecule would be made up of heavy and light. Okay? So, what is DNA replication? It is how the name sounds, replicating our DNA or making more copies of it. Where does this occur? We should all be screaming out at the computer now saying nucleus, but that's not the only place. So bacteria doesn't have a nucleus. So where does it occur in bacteria? In their nucleoli. And when does this occur? And we should all know the answer to that as well. So there are 3 types of replication. So before was like the mechanism. So these are types. These are ways that it can occur. So the first one is in bacteria, they do something called theta. It's named because if you look here and you're familiar with the Greek letter theta, it resembles a theta. So this is their circular DNA. Open it up, bidirectional, and it resembles a theta. And in the end, they would have to cut this and repair so that they get 2 individual molecules. You have rolling circle. So this is what our F factor does. This is like resembles a piece of tape. So you like nick the DNA, because remember it's still double stranded. Use one strand to replicate off as you would a piece of tape. So go around and kick it off. Then you have eukaryotic linear replication. So this is what occurs in eukaryotes who have linear chromosomes. And you have multiple origins of replication that are bidirectional. Okay, how do we feel about that? So that is how we figured out semi-conservative and the 3 ways that replication is carried out in different organisms.